Okay, we're now going to cover Unit 3, Part 1B. Please ignore the birds chirping, but it is definitely better than the dog barking. So, right now we are going to cover from the middle of page 2 and pretty much page 3 on your outline. Last time we talked about things like invasion of tissues versus cells, and there are certain aspects to, for example, bacteria, where they release things that go against to help evade and break down the host defenses. So today we're going to talk about exo and endotoxins. This picture should be intimately familiar to you because you should in fact expect to see this on your exam. So when you have some type of toxin production, it produces toxigenicity. These are usually enzymes and they're soluble proteins that's then secreted into the host. Some of them are more lethal than others. If you have something like a neurotoxin, it impacts the nervous system. So there are many different kinds, and you can look at this in the middle of page 2 on your unit outline. So we're going to talk about the difference between exo and endotoxins. You need to know what is located in a gram-positive or a gram-negative or possibly both type of cell. Please think back to when we covered the cell wall, and in a gram-negative, you had an LPS. That was that outer layer when we were talking about the cell wall. And within the LPS, which is designated as orange in this picture here, these little red dots around the edge of the LPS are actually embedded, and these are known as endotoxin. So if endotoxin is an LPS, then it should be known. This guy right here is endotoxin, and these are only found in gram-negative bacteria. So what occurs is the endotoxin eventually releases, which then impacts negatively the host cell that this bacteria has invaded. Endotoxin, once that is released, anytime you take something out of a membrane, it causes it to burst and pop. So therefore, once you have endotoxin release, it will in fact kill off the bacteria that released it. On the other hand, down here, you have exotoxin. Exotoxin is found in both gram-positive and gram-negative cells. And what you should notice in the picture is it is released, or the word is secreted, out of the bacteria. Therefore, it's not like the endotoxin that's embedded and actually dies off once it's released from the LPS. This is a protein, and it is inside of this toxin is inside of the bacteria and secretes out without killing the cell that's actually secreting it. I'd also like to mention that with endotoxin, again, the lipid A portion of that LPS is what is actually infectious. When these toxins are secreted into the host, a slew of things can occur, including an inflammatory reaction, which is fever, a hypotension. There can be issues with cardiac output being decreased, shock. It can cause infections. It can cause symptoms, diarrhea, for example, if it's an endotoxin in the intestines. I would like to give you an example of an endotoxin is hemolysin. Heme should make you think of blood. We covered this pretty well in lab by now. So remember that you have the blood agar plate, and the differential property is hemolysis. There's beta, alpha, and gamma. Beta is the complete breakdown of blood cells. So when you were to hold up that plate, you can see a clearing because that toxin has broken down the blood cells. So I got a little ahead of myself talking about the LPS. So again, with endotoxin, the organism that's releasing it is going to die at that point because it's embedded in that outer membrane, or specifically in the LPS. This is the lipid A portion, is where the toxin is. This is much weaker when you compare it to an exotoxin, because if you remember what we just talked about, an exotoxin secretes and doesn't kill off the cell that is secreting that toxin. So the LPS does cause many pathophysiological changes, and these are ones that I mentioned in the previous slide. So now what we're going to do, which I mentioned in the outline discussion, we're going to look at, again, portals of entry, the number of invading microbes, which is ID 50, as well as adherence. We're going to talk about how these different toxins and other components of bacteria or microbes in general penetrate and evade the host cells, then how they damage them, and finally, portals of exit. We're going to cover this for all 5 microbes, but please remember that everything we've been talking about to now has been bacterial related, so we still have viruses, helminth, fungi, and protozoa left. As I mentioned, this was all written out for you, not only on page 3 of the outline, but it's also in the slides to come. So viruses act quite differently than bacteria. Remember, as we talked about in Unit 1, there are many different kinds of viruses. They don't follow a typical pattern of any sort. So viruses go through different life cycles. Their portal of entry is exactly the same as bacteria, but what they do for adherence is they work with receptors. So the virus attaches to receptors on the target tissue of the host cell, and again, that is for their level of adherence. They do an invasion, not a penetration like you've seen with bacteria. And again, this has to do with the fact that they are attaching to receptors. They do, in fact, cause many different CPEs. CPE stands for cytopathic effects. So cytopathic effects are the different type of negative effects that these viruses can have on the host. They can kill the cell, they can change how they look, and they can transform them or change how they work. So that is the chart for viruses. Here's a couple pictures and examples of viruses. This is specifically herpes virus. So again, for viruses, they have adherence and must invade. They do this based on receptors as opposed to the different types of toxins and other things that occur with bacteria. And the cytopathic effects are direct damage by killing off the target tissue and other kinds of hypersensitivities, which is another word for allergic reactions. So now we want to look at this chart in regards to fungi, protozoan, and helminths. The key terms and things that I need you to pay attention to are written out for you in the slides to come. So first we're going to take a look at fungi. Many fungal infections are on the skin. A simple one that most people are familiar with but may or may not know is a fungal infection is athlete's foot. So if any of you have it or have a family member that has athlete's foot, you know it is incredibly difficult to get rid of. It's something that you treat over and over again by washing the feet, drying carefully, using powders and ointments. But fungal infections are tough to kill off. If you've heard of cocci, which is valley fever, that is also fungal spores being inhaled. So it's a respiratory illness, and portal of entry would be respiratory. Again, that is difficult to get rid of and quite chronic. So how do fungi work? Well, they invade cells. Now remember, bacteria tend to invade tissues. They also release toxins, which is very similar to what we talked about with bacteria. The example that I gave you was endo and exotoxin, and they cause allergic reactions, which are also referred to as hypersensitivity. As I mentioned, valley fever, often you have spores that are ingested that then cause problems within the host. These pictures are examples of fungal infections gone seriously wrong. This is actually a foot in case you can't recognize it. So for protozoa, it does a full tissue destruction. So it isn't just a simple cell invasion, and it isn't just a tissue colonization, it is a full tissue destruction. Often protozoa have different types of asexual lifestyles. They are transmitted by mosquitoes at times, which are vectors, and many protozoan infections are waterborne. An example of a protozoan infection is Giardia lamblia. This is also known as traveler's diarrhea or Montezuma's revenge. So for helminths, helminths also have the ability to release toxic waste, and they don't just invade cells and they don't just colonize in tissues, they also destroy tissue, just like with a protozoan infection. These can be foodborne, for example, pork that is undercooked and consumed by the primary host, We'll talk a little bit more about this in later chapters.